Good morning, everyone. I am Madhurika, lecturer in Botany, TTWRDC Sagarati. It's my pleasure to share this video lecture with you all. In today's lecture, I would like to talk about the fundamental thing of cell biology, that is ultrastructure of cells. Especially, I'm going to talk about the ultrastructure of plant cell as we are studying botany. Everyone knows about the cell, but in today's session, I'm going to give a clear-cut idea about the cell organelles and its function in very depth and in brief manner. So let us begin our today's session. First of all, I would like to give the introduction of plant cell. Not only plant cell, all the living organisms are made up of cells. So from unicellular organisms to multicellular organisms, every organism should be made of cells. That's why the cell is become the structural and functional unit of all living organisms. If you see the structure of a plant cell here, which is visible very clearly, the outermost layer of the plant cell is called cell wall. As it is the outermost layer, it is giving protection and it is giving certain shape to the cells. And next to the cell wall, there is inner membrane called cell membrane, or also known as plasma membrane. And the cell wall and plasma membrane along with the inner cytoplasm commonly called as protoplast. And the protoplast, presence of protoplast is the major difference between a living cell and non-living cell. And remember, basically cells are of two types, whether it is living cell or non-living cell. A living cell is with cytoplasm and protoplast, and a non-living cell lacks protoplast. And coming to the hierarchy of cell, there are eukaryotic cell as well as prokaryotic cell. The plant cells are called eukaryotic cells that I have mentioned here. What is the meaning of a eukaryotic cell? Means the eukaryotic cell is the cell which is having all the cell organelles in association with the nuclear membrane, nucleolus, and well-defined nucleus. Whereas the prokaryotic cells lacks the well-defined nucleolus, nuclear membrane, and may contain circular chromosome. So we can differentiate eukaryotic cell with prokaryotic cell with taking example as bacterial cell as well as plant cell. Next, the subcellular organelles means the cells, the cell organelles which are present in the cell are called as subcellular organelles. All the plant cells contain many subcellular organelles. And one of the important characteristic feature of plant cell is plasmodesmatal connections. The plasmodesmatal connections are the connections through which the transportation of materials take place between two cells or among many cells. That means adjacent plant cells can continue their connection with plasmodesmatal connections. This is the little bit about the plant cell. And let us see in depth structure of plant cell wall and plant's various cell organelles. Right. So in this slide, we are going to study about the cell organelles. The subcellular organelles which are present inside the cell are called cell organelles. If you see clearly, there are many cell organelles that have well labeled here. See. So in this session, in this slide, I would like to give clear-cut idea about the various cell organelles. Right. So from outer to inner side, I am telling. See, the outermost layer of this cell is called cell wall. As it is the outermost layer, it giving certain shape and protection to the inner parts. Next, coming to the inner side, there is the membrane called plasma membrane, which is also known as plasma lemma. This is just present inner to the cell wall. And this is the gel-like structure. This entire area is called as cytoplasm. As we are mentioned earlier, the cytoplasm is the living matter that is present inside the cell. And remember, the cytoplasm is the platform where all the subcellular organelles are present. Right. So in the cytoplasm, all the subcellular organelles called cell organelles will be present. There are many cell organelles like this is a chloroplast, which is the cell organelle useful for photosynthesis. This is mitochondrion, which is the cell organelle useful for respiration. And these are Golgi complex, and this is endoplasmic reticulum, and these are ribosomes. This is peroxisome. So these are all the various cell organelles that are associated with various functions. In this session, I'm going to discuss in depth and very brief introduction regarding all the subcellular organelles one by one in the next slides. Let us go towards the next slide again. See? So once again, I'm repeating the cell organelles that are present in the plant cell are cell wall, plasma membrane, cytoplasm, nucleus, mitochondrion, chloroplast, Golgi complex, endoplasmic reticulum, peroxisomes, vacuoles, ribosomes, etc. Yes. So first, I would like to talk about the primary and the outermost layer of cell, that is cell wall. Clearly, we can see the structure of cell wall here. The cell wall is the outermost layer, and presence of cell wall is the important characteristic feature of the plant cell. If you compare a plant cell and an animal cell, the plant cell will be having the cell wall, but in animal cell, there is no cell wall. So the animal cells are lacking cell wall, but the plant cells are having cell walls. This is the major difference between the plant cell and the animal cell. And just like plant cell, many other organisms, many other living organisms are also having plant cell, but based on the wall material, we can differentiate the plant cell from other organisms. Okay, look at the wall structure, cell wall structure. The cell wall is made up of cellulose, hemicellulose, and pectin. So cellulosic cell wall is the characteristic feature of plant. Coming to the bacteria is also having cell wall just like plant cell, but the wall material in the bacterial cell is not at all cellulose. It is peptidoglycol layer. And fungi is also made up of cell wall. That means fungal cell also having cell wall, but the wall material is something other than cell wall, something other than cellulose, that is chitin. So simply we can say the cell wall may present in many living organisms, but the living organisms can be differentiated based on the cell wall component. So cell wall of plant cell is made up of cellulose, and cell wall of bacterial cells are made up of peptidoglycan layer, and the cell walls of fungal cell wall are made up of chitin. So this is the clear-cut difference between the cell wall of fungi, bacteria, and plant cells. And so the cell wall of plant is made up of majorly with cellulose, and some little amount of hemicellulose and pectin is also there. As it is, cell wall is the outermost layer. It provides protection and shades to the cell. And one of the characteristic feature of plant cell is presence of pit area. The pit areas, through this pit areas only, the plasmodesmatal connections will take place. And the structure of cell wall contains 3 layers. The 3 layers of the cell wall are middle lamellum, primary cell wall, and secondary cell wall. The middle lamellum is the cementing layer between 2 adjacent cells, and the primary cell wall is present in all the plant cells, but the secondary cell wall will be seen only in case of mature plant cells only. And remember, one important competitive examination questions are, the primary cell wall is elastic. So means the cellulosic, it is made up of majorly with cellulose, and the primary cell wall is elastic, whereas secondary cell wall is inelastic. That means it cannot be elastic. What is the reason? Means the primary cell wall contains only cellulosic wall material, and the secondary cell wall, as it is present in matured cells, this secondary cell wall should provide additional strength. That's why the secondary cell walls are made up of lignin, suberin, like these hard materials. As the secondary cell walls are made up of hard materials, the secondary cell wall is not at all elastic, but the primary cell wall is elastic, and this primary cellulosic cell wall is permeable for all substances. So now we can see the clear-cut diagram of cell wall structure. Yes, see this. This is the cell middle lamellum and this is called primary cell wall. This is the plasma membrane of the cell. Here, this is not a matured cell. That's why there is no secondary cell wall here. Just before I have said that secondary cell wall will be present only in case of matured cells. As it is the young cell, only middle lamellum, primary cell wall is there. And the plant cells are majorly made up of cellulose, hemicellulose and pectin. This is about the cell wall. Let us go towards the next cell organelle. The next cell organelle is plasma membrane. Just inner to the cell wall, there is the plasma membrane. The plasma membrane is the limiting membrane. It forms the boundary for cytoplasm and the plasma membrane is also called as plasma lemma. There are various models that were proposed for the plasma membrane structure. Now, the universally acceptable model is fluid mosaic model, which is proposed by Singer and Nicholson. But prior to that, there are many models that have revealed the structure of plasma membrane. Not at all, they are fact. But in view of competitive examination, I would like to present the various models of plasma membrane proposed earlier to Singer and Nicholson model. Let us have a glance. First of all, the first proposed model for plasma membrane is called sandwich model. This sandwich model is proposed by Daniel and Davson. This is the sandwich model. Here, the lipid bilayer is present like this. These are the proteinaceous molecules. So, in the summary of Daniel and Davson's model, the sandwiched lipid molecules are present in the proteinaceous structure. This is the summary. And coming to the next model proposed for plasma membrane is unit membrane model. The unit membrane model is proposed by Robertson. The unit membrane model is also called as greater membrane model. This is also complementary, but Robertson revealed one fact, that is all the membranes, whatever the membrane, that means it may be plasma membrane, it may be nuclear membrane, it may be chloroplast membrane, whatever the membrane, all the membrane structure has similar in nature. This was the fact that has proved by Robertson. And the micellar model of plasma membrane is given by Hiller and Hoffman. And finally, the universally accepted model of plasma membrane was proposed by Singer and Nicholson. This is called fluid mosaic model. Let us have a glance at fluid mosaic model of plasma membrane. Plasma membrane of fluid mosaic model. This is the structure of plasma membrane, which is having fluid mosaic model. All the plasma membranes, means all the membranes or plasma membranes are lipoproteinaceous membranes. That means they are made up of lipids and proteins. That's why the lipoproteinaceous is the nature of plasma membrane and all the limiting membranes. And remember, the membranes are always semi-permeable. They are not going to allow all the substances into and out of the cell. They will allow only certain and selective substances into and outside of the cell. This nature makes the plasma membrane semi-permeable. And if you see the structure of fluid mosaic model of plasma membrane, these are the bilayered lipid molecules are present in the center. And these are blue colored ones are called proteins. These proteins are randomly arranged in the mosaic pattern in the sea of lipids. So Singer and Nicholson has shown this lipid as the sea and these protein molecules as the iceberg. So as they are arranged as the integral or integral parts of the proteins in the form of lipids, this is called fluid mosaic model. This fluid mosaic model by Singer and Nicholson explained as the proteins are icebergs in the sea of lipids. This is the structure of plasma membrane. Not only plasma membrane, all the limiting membranes will be having the same structure and same function. All the membranes are lipoproteinaceous and all the membranes are semi-permeable in nature. And these two and there are many functions of plasma membrane. The major functions of plasma membrane is pinacocytosis, that means cell drinking. If any amino acid or liquid particles are trying to enter the plasma membrane, simply it will swallow. This type of drinking is called as phagocytosis, or also called as cell drinking. And phagocytosis is also another function of plasma membrane. Any larger sized particles or any type of vesicles are trying to enter to the plasma membrane, directly it will surround that particular vesicle and swallows it. This is called phagocytosis. So simply, these are all the plasma membrane structure and its functions. Now, let us go towards the next cell organ. Cytoplasm. The cytoplasm is the platform which is having all the subcellular organelles. So, majorly, the cytoplasm will be having watery fluid or gel-like substances, and it is enzyme-rich, protein-rich. It contains all the amino acids, proteins, nutrients, because most of the metabolic reactions are going to take place in the cytoplasm. That's why. And so, the cytoplasm is the living matter of the cell. And I have already told earlier, there are two cells which is called living cell and non-living cell. A non-living cell lacks cytoplasm and a living cell will be having cytoplasm. So, most of the enzymes and proteins are present in the cytoplasm that are useful for various metabolic reactions, and it is the platform for all the cell organelles should be present. Next. So from this onwards, I would like to share there are many subcellular organelles in the cell. Some of them are having double membrane structure, some of them having single membrane structure. There is one cell organelle which does not having any kind of membrane. So in this sequential manner, in this respected manner, I would like to tell the class. Right. So first, I would like to speak about the cell organelles which are bounded by two membranes. So the nucleus is the membrane which is having two membranes. So the nucleus is bound by two membranes. This is the structure of the nucleus. Have a look here. This is the outer nuclear membrane and this is the inner nuclear membrane, and these are small pores. So the nuclear membrane is not at all continuous. These nuclear membranes will leave very small pores. These are called nuclear pores. So next, inside of nucleus, there is the matrix-like substance. This is called nuclear sac. And this thread-like structure that is present in the nucleus is called chromatin. When the chromatin undergo condensation, they become chromosomes. Inside the chromosomes, we know very well the DNA will be present. And this round ball-like structure that is present inside the nucleus is called nucleolus. So simply, in the entire structure of nucleus, in the ultrastructure of nucleus, how many parts are there? Simply, there are just 4 parts. What are the 4 parts? Let's recapitulate again. The nuclear membrane, which is having outer nuclear membrane and inner nuclear membrane with spores. And the matrix that is present inside the nucleus is called nuclear sac. And the chromatin or chromosomes, if it is in uncondensed form, that is called chromatin thread. If it is undergo condensation, they will form rod-shaped bodies called chromosomes which contain DNA. And the another rounded body, deeply stained dark body is called nucleolus. This nucleolus having special function, this will synthesize the ribosomes. That's why the nucleolus is also called as ribosomal factory. And see, this is about the nucleus. So having all these functions, the nucleus is called as cell brain or master control of the cell. So this is the ultrastructure of nucleus. Let us go towards the next cell organelle, that is mitochondria. This is the diagram of mitochondria. Let us see what are the structures that are present inside the mitochondrial structure. Right. First of all, the mitochondria is also bounded by two membranes. Have a look at this diagram. This is the outer membrane of mitochondria and this is inner membrane of mitochondria. If you observe clearly, the outer membrane of mitochondria is smooth. It does not have any foldings. But if you see the inner membrane of mitochondria, it is having infoldings. These infoldings of inner membrane of mitochondria is called as crest. or cristae. These are cristae. And see, in between these foldings, there is the matrix-like substance or enzyme-rich and protein-rich structure substance is there. This is called matrix of mitochondria. And these round-shaped particles are called oxysomes, are also called as F0F1 particles. These are also known as ATP synthase. As they are associated with ATP formation, they are called as ATP synthase also. This is the structure of mitochondria. Okay. What is the function of mitochondria? The mitochondrion is the cell organelle that is associated with respiration. All the glycolysis, Krebs cycle, electron transport chain, everything about respiration occurs in the mitochondria. And these mitochondria undergo respiration and forms energy. And this energy will be stored in the mitochondria. That's why the mitochondria are popularly known as powerhouses of the cell. And it is also called as cell within the cell. Why? Because it is having the prokaryotic cell structure, as it is having its own DNA that is called mitochondrial DNA, and it is also having its own ribosomes. They are called 70S type of ribosomes. And with this all its own DNA and ribosomes, with this all enzymes and proteins, it is having one prokaryotic structure. That's why the mitochondrion is also called as cell within the cell or prokaryotic cell within the eukaryotic cell. And the major function of mitochondria is associated with respiration and its stored energy. That's why it is popularly known as powerhouses of the cell. So this is the structure of mitochondrion, which is also bounded by two membranes. Next. The next cell organelle is chloroplast. Right. This is the structure of chloroplast. If you see the structure of chloroplast, the chloroplast is also the cell organelle which is bounded by two membranes. The chloroplast membrane, that is outer plastidial membrane, and the inner one is called inner plastidial membrane. And the space is called, the space between two membranes is called periplastidial space. And if you see clearly, inside the chloroplast structure, the chloroplast contain the matrix-like substance. This entire gel or matrix-like substance that is present inside the chloroplast is called as stroma. And this stroma contain the sac-like structures. These are called thylakoids. Here, clearly we can see all these rounded green color structures are called thylakoids. These sac-like structures, sometimes they are forming stalk of the thylakoids, sometimes they are singular. So, if the sac-like structures called thylakoids, these thylakoids are forming. If these thylakoids are forming stalk of the thylakoids, that is called grana thylakoids. And if they are singular, they are called stroma thylakoids. In grana thylakoids, stroma thylakoids, there is the differentiation of photosynthetic pigments and photosystems. In grana thylakoids, there is various kinds of photosystems, whereas in stroma thylakoids, there is another type of photosystem. This is the difference. And as like mitochondria, the chloroplast is also having its own DNA. This is called cDNA or cpDNA. That is chloroplast DNA. And it is also having its own ribosomes. They are called 70S type of ribosomes. And everyone knows about the function of chloroplast. That is nothing but photosynthesis. What is photosynthesis? Photosynthesis is nothing but by using light, the plant can synthesize its own food material as carbon dioxide and water as ingredients. This is the photosynthesis. We know very well about the photosynthesis. So simply, once again, I am recapitulating the entire structure of chloroplast. The chloroplast is the cell organelle that is useful for photosynthesis. And if you see the structure of chloroplast, it is also bounded by two membranes, that is outer membrane, inner membrane, and the gel-like or matrix-like substance that is present in the chloroplast is called stroma. In the stroma, sac-like structures are there, they are called thylakoids. The thylakoids are of two types. If they are stock, if they are forming a stalk, then they are called thylakoids. If they are singular, then they are called stromathylakoids. And there is the difference between grana and stromathylakoids in having photosystems, photosystem 1 or photosystem 2. So just like mitochondria, it is having its own DNA that is called chloroplast DNA. It is having its own ribosomes. They are called 70s type of ribosomes. So simply in the cell, there are 3 cell organelles which are bounded by two membranes. That means the double membrane bound cell organelles are 3. What are the 3 double membrane bound cell organelles? Let us recapitulate. First, the master control of the cell or cell brain, that is nucleus. Nucleus is bounded by 2 membranes, outer nuclear membrane, inner nuclear membrane, and mitochondria is also bounded by 2 membranes, that is outer mitochondrial membrane, inner mitochondrial membrane forming cristae. As well as these two, the chloroplast is also another cell organelle which is also bounded by 3 membranes. So let us recapitulate all these 2 membranes bound cell organelles function. The nucleus will be contain genetic material and it controls the entire cell. That's why it is called cell brain. The mitochondria is associated with respiration and it stores energy. That's why it is called powerhouse of the cell. The chloroplast is the cell organelle that is associated with photosynthesis. That's why it is called as photosynthetic cell organelles. So with this, we have completed the cell organelles which were bounded by 2 membranes or double membrane bound cell organelles we have completed. Let us go towards the single membrane bound cell organelles. Next. The next single membrane bound cell organelles, that is peroxisome. First of all, I would like to talk about the peroxisome. Remember, the peroxisomes are the cell organelles. These are bounded by single membrane and they are present only in plant cell. The peroxisomes are absent in the animal cell. And if you see, what is the function of peroxisome? The peroxisome is the important and characteristic feature of plant cell. There is this special pathway which is called as glycolate pathway in photorespiration. In glycolate pathway, the peroxisome is the major cell organelle that should be operated in the C2 cycle called glycolate pathway. So simply we can say without peroxisome, there is no glycolate pathway in plant cells. And this peroxisome majorly contain catalase enzyme or catalase-like detoxification enzyme. What is the function of catalase enzyme? The catalase enzyme is the enzyme which detoxifies the hydrogen peroxide, toxic one, into water and oxygen. So in this manner, the detoxification of hydrogen peroxide will be taken place in peroxisome. As the peroxisomes are rich in catalase like detoxification enzymes, this cell organelle is associated with detoxification of various toxic substances. And this peroxisome is also site of fatty acid breakdown in the plants. So with this peroxisome structure and function, in brief manner, it is completed. Next cell organelle, that is Golgi complex. Yes, what is a complex? Simply we can say the complex is nothing but which is having more than one type of cells or more than one should be present in the complex. So Golgi complex is the cell organelle which is having 3 types of cells, or it is an association of 3 different kinds of cells. See, the 3 different kinds of cells of Golgi complex are, here we can see the cisternae, tubules and vesicles. These 3 are the different types of cells that are present in the Golgi complex. As the Golgi, this cell organelle is made up of 3 different types, that is called as Golgi complex. The Golgi complex is the cell organelle. This is associated with secretion of lipids and secretion of enzymes and secretion of hormones. So This is majorly is with lipid concentration. That's why the Golgi complex is also called as lipochondrial. This is one of the important competitive examination question which is having the scope to ask in various competitive exams. And it is also called as idioso, and it is also called as Baker's body. And one of the important function of Golgi complex is the Golgi complex is a cell organelle which is very much useful for cell plate formation method. Cell plate formation method is the method which can synthesize the plant cell wall. Earlier we have discussed about the plant cell, right? So the plant cell is the outermost layer which is giving shape and protection to the inner parts. So the Golgi complex is a cell organelle which is one of the important cell organelle that should synthesize the cell wall. And next, coming to the next cell organelle, that is vacuoles. So one of the important characteristic feature of a plant cell is presence of vacuoles. Remember, just like peroxisome, the vacuoles of the cell organelles, they are also present only in plants. So simply we can say the vacuoles of the cell organelles, they are absent in the animal cell. So the presence of vacuoles is characteristic of plant cell. See, here we can see the structure of vacuole clearly. Then vacuole is something called as very special cell organelle. Why? Because not only present in the cell, this is not the reason. Every cell membrane should be synthesized first, and later it has any damage, they undergo repairing mechanism. But there is one and only membrane which is undergo de novo synthesis, means from first it has to synthesize, that is the vacuolar membrane. And remember one more interesting fact about the vacuole is every membrane having, for example, nucleus. Nucleus is bounded by membrane. That membrane which is present in the nucleus, we are calling it as nuclear membrane. Just like this, mitochondria is also bounded by membrane. That membrane we are calling it as a mitochondrial membrane. Chloroplast is also bounded by membrane. It is called as chloroplast membrane. But there is the special name for one and only cell organelle, that is vacuole. The membrane of the vacuole is having special name which is called as tonoplast. So remember, this is also a competitive examination question. The tonoplast is the membrane of the cell organelle. Then we have to say the cell organelle name is vacuole. So the vacuolar membrane is called source, tonoplast. And in mature cells, mostly the vacuoles are present. And what is the function of vacuoles? The vacuoles of the cell organelles, they are acts like a storehouses. They are going to store various detoxifying substances or any other materials, wrappers, crystals, whatever the material that may be stored in the vacuoles. As the vacuoles of the cell organelles, they are useful for storing of various substances. The vacuoles are called as a repository of the cell. Even coming to the camp cycle, even the malate is also going to store in the vacuole at the time of life. So just like this, the vacuoles of the cell organelle, they are useful for the storage of various substances. That's why the vacuole becomes the repository of the cell and the storehouses of the cell. Along with the storage of various substances, there is one special function that is associated with vacuole. The vacuole is also useful to conduct osmoregulation, which is known as it will maintain water balance inside the cell. So osmoregulation can be also one of the important function of plant vacuoles. Okay. Let us see next cell organelle. Yes. The next cell organelle, that is endoplasmic reticulum. The endoplasmic reticulum forming the cytoskeletal system. Let me give you a small example regarding the importance of endoplasmic reticulum. We are human beings. Our skeletal system is interacted and supported by muscle system. If we does not have our skeletal system, what will be our shape and what is our nature? We will like a clot, right? So just like that. In a human body, how the skeletal system providing mechanical strength to the human body, this kind of mechanical strength in the plant will be, in the plant cell will be supported by endoplasmic reticulum. So simply the endoplasmic reticulum inner side of the plant cell will form in the cytoskeletal system. So one of the important cell organelle of the plant cell is endoplasmic reticulum. Right, see. The endoplasmic reticulum is of two types. That is smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth endoplasmic reticulum is one type of endoplasmic reticulum which is also called as SARs, and the rough endoplasmic reticulum is also one type of endoplasmic reticulum which is associated with ribosomes. So the major difference between the smooth endoplasmic reticulum and rough endoplasmic reticulum is the rough endoplasmic reticulum is associated with ribosomes, whereas the smooth endoplasmic reticulum does not associated with any kind of ribosomes. Yes, here we can see the structure of endoplasmic reticulum here. And remember one important thing, always the endoplasmic reticulum has its origination from outer nuclear membrane to nuclear pores. Earlier we have studied about the ultrastructure of nucleus. As the nucleus is bonded by two membranes, the outer nuclear membrane is continuous with endoplasmic reticulum. This diagram is depicting us very clearly. This outer nuclear membrane is giving rise to endoplasmic reticulum. Here we can see this is the rough endoplasmic reticulum as it is having ribosomes like this. These granule-like structures are called ribosomes. So this is rough endoplasmic reticulum. But see these tube-like structures, there is no any kind of granules here. So without ribosomes, this endoplasmic reticulum is present. That's why this is called smooth endoplasmic reticulum. So the functions of endoplasmic reticulum is it forms intracellular transportation channel, and it is also associated with the detoxification of drugs, and it is useful for the formation of Golgi complex, and it forms the cytoskeletal system inside the plant cell to give the support. So this is about the endoplasmic reticulum. Next, come to the next cell organelle. Right. So next cell organelles are ribosomes. This is something special kind of cell organelles. The ribosomes are called universal cell organelles. Let us see why ribosomes are called as universal cell organelles. See. The ribosomes, just like endoplasmic reticulum, there are also two types. There are 70S type of ribosomes and 80S type of ribosomes. There are two types of ribosomes. The 70S type of ribosomes are usually present in prokaryotic organisms, whereas the 80S type of ribosomes are present in eukaryotic organisms. This is the structure of 70S type of ribosomes as well as 80S type of ribosomes. And the ribosomes, 70S, we are calling ribosomes as 70S, 80S. This S stands for Swedberg unit. And see, if you see the prokaryotic 70S type of ribosomes, this will be having again three types of subunits. The ribosomes having two types of subunits. They are called larger subunit and smaller subunit. The 70S type of ribosomes will be having larger subunit as 50S and smaller subunit as 30S. This is the 70S type of ribosomal subunits. And in turn, the 50S type of larger subunit is having 5S RNA, 23S RNA, whereas the 30S type of smaller subunit having 16S RNA. And Just like this, the eukaryotic ribosomes are also having two units, the larger subunit and smaller subunit. The larger subunit of 80S type ribosomes having 60S and smaller subunit is 40S. The 60S subunit is having 5S RNA, 5.8S RNA and 28S RNA. This is the integral parts of 60S subunit, and the 40S subunit is having 18S RNA. So simply we can say the ribosomes having larger subunits and smaller subunits, and there are 70S type of ribosomes and 80S type of ribosomes. The 70S type of ribosomes are present in prokaryotic ones. The 80S type of ribosomes are present in eukaryotic organisms. And these are the subs and remember, there is no membrane. That means the ribosomes are membraneless cell organelles. What may be the reason why there is no membrane for these cell organelles? As they are in the form of two subunits. See, remember, the ribosomes are not always in associated form. The larger subunit will be somewhere in the cell and smaller subunit will be somewhere in the cell. As they are not associated always, there is no possibility of a membrane. But when the protein synthesis is going, at that time only, the larger subunit and smaller subunit will come closer and undergo protein synthesis. So this is the reason why ribosome does not have a membrane. So in the entire cell, there is one and only membraneless cell organelle, that is ribosomes. And why we are calling ribosomes as the universal cell organelle? See, as we are studied earlier, there are two types of cells, that is eukaryotic cell and a prokaryotic cell. The eukaryotic cells are the advanced cells and prokaryotic cells are somewhat primitive kinds of cells. In primitive kinds of cell, only 70S type of ribosomes are present. But in eukaryotic cells, 80S types of ribosomes are present. So, whatever the cell, whether it is prokaryotic or eukaryotic, the one and only cell organelle that is present is ribosomes. In prokaryotic cells like bacteria, only 70S type of ribosomes are present. Mitochondria, Golgi complex, endoplasmic reticulum, all these cell organelles will be absent. So, the one and only cell organelle that is present in prokaryotic as well as eukaryotic organisms is only ribosomes. So, because of this reason, the ribosomes are called as universal cell organelle, as they are present in prokaryotic organisms as well as eukaryotic organisms. Next, the function of ribosomes is protein synthesis. The protein synthesis machinery always revolves around the ribosomes. So that's why the ribosomes are popularly called as protein factories, as they are associated with protein synthesis. So this is about the various cell organelles that were present in the cell. Once again, I would like to give the recapitulation of entire today's class. Today's class is about the ultrastructure of cell wall, ultrastructure of plant cell. The plant cell is having various cell organelles. The outermost layer of plant cell is cell wall, which is a cellulosic cell wall, and it is giving protection and shade to the inner parts. And inner to the cell wall, there is plasma membrane. The plasma membrane is the cell organelle, which is the boundary for all the cell organelles. And it is lipoproteinaceous. It is semi-permeable in nature. Next. The well-accepted model of plasma membrane is fluid mosaic model, which is proposed by Singer and Nicholson. And the next cell organelle is called cytoplasm. The cytoplasm is the matrix or gel-like substance which is enzyme-rich and protein-rich. It is forming the platform for all the subcellular organelles, and it is associated with various metabolic activities. And the next cell organelle is nucleus. This nucleus is called as cell brain or master control of the cell. Master control of the cell. Next, mitochondria. This mitochondrion is associated with respiration as it stores energy. This is called as powerhouses of the cell, and as it is having its own DNA and ribosomes, it is called cell within the cell. And the next cell organelle is chloroplast. The chloroplast is the cell organelle that is associated with photosynthesis, which provides the food for all living organisms. That's right, which makes plants autotrophic. This is also bounded by two membranes. And next, coming to the single membrane bound enzyme, single membrane bound cell organelles, that is peroxisome. Peroxisomes or single membrane bound cell organelles, they are associated with glycolate pathway and detoxification of toxic substances. And next, single cell membrane bound cell organelle is Golgi complex. The Golgi complex is the cell organelle which is very important in cell plate formation method, and it is useful for the secretion of lipids. And that's why it is also called as lipochondrion or Baker's body. Next, the next important cell organelle of the cell is vacuole. This is one of the peculiar or characteristic of plant cell, that is called vacuole. The vacuole having special features. The name of the vacuolar membrane is called tonoplast. And the vacuole sap contain various digestive enzymes or detoxifying enzymes, as it is useful for storage of various cell organelles. This is called repository of the cell, and in addition to this, is also conducts osmoregulation. The next cell organelle is called endoplasmic reticulum. The endoplasmic reticulum is two types. It is rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is associated with ribosomes, whereas smooth endoplasmic reticulum is not associated with any kind of ribosomes. And the function of endoplasmic reticulum is it forming the cytoskeletal system and it forming the intracellular transportation channel. Next cell organelle is ribosomes. Finally, we come for ribosomes. The ribosomes are called universal cell organelles. These universal cell organelles or membraneless cell organelles, they are associated with protein synthesis. That's why they are called protein factories of the cell. Why they are not having membranes means because they are in the form of larger and smaller subunits. So at the time of protein synthesis only they are going to bind. That's why the membrane is not applicable for this. And there are two types of ribosomes. Always the 70th type of ribosomes are present in prokaryotic organisms and 80th type of ribosomes are present in eukaryotic organisms. This is the eukaryotic and prokaryotic organisms differentiation. So these are the various cell organelles that were present in the plant cell and about their function. So I would like to conclude today's topic with the two types of cells called prokaryotic cells and eukaryotic cells and what is their difference. So all the unicellular to multicellular organisms are made up of cells. The cell organelles are different in case of eukaryotic cells and prokaryotic cells. The prokaryotic cells are primitive cells like bacteria. The eukaryotic cells are somewhat advanced cells like plant cells. So the major difference between eukaryotic cells and prokaryotic cells is in eukaryotic cell, the very well organized, well defined nucleus is present. But in prokaryotic cell, the nuclear membrane and nucleolus is absent and the circular chromosomes are present. And one of the important differentiation between prokaryotic and eukaryotic cell is in eukaryotic cell, the 80th type of ribosomes are present like this. And in prokaryotic cell, the 70th type of ribosomes are present. And one more important character of eukaryotic and prokaryotic cell is the eukaryotic cell will be having all the cell organelles which we have discussed till now, but A prokaryotic cell does not consist of any other cell organelle except ribosomes. So simply we can conclude the eukaryotic cell will be having well-defined nucleus and 80s type of ribosomes. But the prokaryotic cells does not have well-defined nucleus, means nucleus and nuclear are absent, and it contains 70s type of ribosomes. This is the prokaryotic and eukaryotic cell. So, coming to the plant cells are called eukaryotic cells. They are very, very advanced types of cells. This is about the plant cell and various cell organelles of the plants. Once again, I would like to show various cell organelles in this session. This is the ultrastructure of plant cell that we have discussed here. This is the cell wall, and inner to this is plasma membrane, which is also called as cell membrane, and this structure is called cytoplasm. And these structures or various cell organelles are present here. The central one, the larger one, is called central vacuole. And these green color structures are called chloroplast, and these pink color structures are called mitochondria, and this blue color structure is called peroxisome, and this light pink color structure is called nucleus. The nucleus is originating and giving rise to endoplasmic reticulum. The endoplasmic reticulum is of two types. Here, the endoplasmic reticulum is not associated with any kinds of ribosomes. That's why it is smooth endoplasmic reticulum. But here, this is having ribosomes. This is rough endoplasmic reticulum. And these are the ribosomes which are known as universal cell organelles. The universal cell organelles are useful for the protein synthesis. That's why they are also called as protein factories. This is entirely about the plant cell. So finally, let us discuss some questions on the ultrastructure of plant cell. How can we differentiate the plant cell from other cell types? So we can answer simply. The plant cell will be having cell wall. The cell wall is made up of cellulose, hemicellulose, and pectin. If it is a plant cell, that should made up of cell wall, and the cell wall material should be cellulose, hemicellulose, and pectin. This cell wall material can be differentiate plant cell from other bacteria, other fungus, any other cell wall material. And what are the characteristic features or what are the cell organelles that are present, that are special for only plant cell? For this question, we will answer like this. The plant cell will be having some peculiar organelles. They are chloroplast first. The chloroplast is the photosynthetic cell organelle that is restricted to only plants. Why? Because the photosynthesis occurs only in plants except some photosynthetic bacteria. So one of the peculiar cell organelle of the plant cell is presence of chloroplast. Next, central vacuole. The vacuoles are also special for only plant cells. These are called repository of the cell or storehouses of the cell. Most of the pigments like anthocyanin, the pink color anthocyanin or red color anthocyanin pigments are stored in vacuoles. So vacuoles are also another cell organelles. They are peculiar and only they are present only in plant cells. And another is peroxisome. The peroxisome is another cell organelle which is also peculiar for plant cell itself. So these three, there are many other glyoxysomes, amyloplast, etc. are also there, but mostly the plant cell peculiar characteristic cell organelles are chloroplast, large vacuoles, and peroxysomes, glyoxysomes, amyloplasts. These are the various cell organelles, and presence of cell wall is also another important characteristic feature of the plant cell. Next question. How can we differentiate a plant cell from animal cell? This is simple. The plant cell is having cell wall, which is made up of cellulose, whereas the animal cell does not have any kind of cell wall. So simply the cell wall will be absent in case of animal cell, but cell wall will be present in plant cell. And another subcellular organelles like chloroplast, peroxisomes, etc. are also going to absent in case of animal cell, but they are present in the plant cell. And the next question, which is the one and only special membrane that is synthesized by de novo mechanism in the entire plant cell? To this question, we have to answer like this. Every membrane, whatever the membrane, it may be nuclear membrane, it may be chloroplast membrane, it may be plastidial membrane, every membrane, if any kind of damage is occurred in the membrane, that should be repaired. The repairing mechanism has to take place. But in plant cell, there is only one membrane which can undergo de novo synthesis, vacuolar membrane, which is having special name, that is called tonoplast. So the tonoplast is the vacuolar membrane which undergo de novo synthesis only. Next, why the cell organelles, ribosomes are called universal cell organelles? To answer this question, the answer is the ribosomes are the universal cell organelles because they are present in prokaryotic organisms as well as eukaryotic organisms. If we see a prokaryotic organisms, all the cell organelles like nucleus, chloroplast, Golgi complex, endoplasmic reticulum, every cell organelle will be absent except ribosome. So only ribosomes are the cell organelles. They are present in the prokaryotic organisms and they are of 70 years time. But coming to the eukaryotes, along with all cell organelles, the ribosomes are also present. So only cell organelle that is present, that is common in prokaryotic and eukaryotic organisms is ribosomes. That's why they are called universal cell organelles. Next, which is called as lipochondria. Which cell organelle is called as lipochondria? Here, we may get it out and we may get into confusion because there are many cell organelles which are associated with lipid synthesis and detoxification of drugs. For example, endoplasmic reticulum is also associated with secretion of lipids, and Golgi complex is also associated with secretion of lipids, and various cytoskeletal system is also associated with secretion of lipids. To answer this question, the right answer is the lipochondria is nothing but Golgi complex because mostly the lipid secretion, major content of lipid secretion is synthesized in Golgi complex. That's why only Golgi complex should be called as lipochondria, or also called as Baker's body, and this is the only cell organelle that is very important for cell wall formation. And final question, which cell wall is made up of 3 layers? Yes, outer cell wall, inner cell wall means primary cell wall, secondary cell wall, and middle lamella. Among the 3, which is the elastic one and which is the inelastic one? This is the competitive examination question. For this question, our answer is the primary cell wall is elastic because the primary cell wall is present in only younger cells. Always the young cells are made up of cellulose. The cellulose is always permeable. So simply the cellulosic cell wall is permeable and elastic. But when cell undergo mature, the matured cells contain secondary cell wall. The secondary cell wall material is always made up of lignin, suberin like hard substances and hard wall materials. So always the secondary cell wall in matured cells will be inelastic. That's why the matured cells, secondary cell wall, not at all elasticity in nature. So this is about the various cell organelles that were present in the plants and various competitive examination questions. They have scope to be asked in the competitive examinations, and various another names and synonyms for this, all the cell organelles. So with this, we should conclude. The cell is the basic structural and functional unit of all living organisms because it is having another many subcellular organelles which is having its own function. So as the entire metabolic reactions, entire metabolism is associated with cell and its cell organelles, the cell is called the basic structural and functional unit of all living organisms. So this is about the cell and ultrastructure of plant cell. So simply, with Sweden and Schwann cell theory, it is clear for everyone that the cell is the structural and functional unit of all living organisms, and always cells arises from pre-existing cells. This manner of arising cells from pre-existing cells is called omnicellular e-cellular. Thank you everyone for giving this wonderful opportunity. Thank you one and all.